Welcome to this lecture on computational biology. Today, we are going to explore two of the most powerful and revolutionary tools in bioinformatics, FASTA and BLAST. Imagine trying to find a single specific sentence hidden within a library of millions of books. Reading every page would take a lifetime. Biologists face a similar challenge when comparing DNA or protein sequences against massive genomic databases. In this session, we will uncover the clever mathematical shortcuts these algorithms use to find genetic matches in minutes rather than years, and understand why they are absolutely fundamental to modern biological research. Let us begin by understanding the sheer scale of the sequence alignment problem. Why do we even need specialized tools? Well, comparing a newly discovered DNA sequence to every existing sequence in a global database requires immense computational power. Historically, scientists used exact mathematical methods that compared every single letter from start to finish. Think of this as trying to find a specific sentence in a massive library by reading every single book cover to cover. It is mathematically perfect, but practically impossible. As biological databases began to grow exponentially, these slow, exhaustive methods became completely impractical for everyday laboratory use. Now that we understand the limitations of exhaustive searching, let us look at the solution. Before 1985, scientists relied on dynamic programming algorithms like Smith-Waterman. While these were mathematically perfect, they were incredibly slow. To keep up with the exponential growth of biological databases, researchers needed a new approach. This is where heuristic algorithms come in. A heuristic method does not guarantee an absolute, mathematically perfect alignment every single time. Instead, it takes a smart, calculated shortcut to find a highly accurate answer very quickly. This shift from perfect but slow to heuristic and fast revolutionized computational biology. This brings us to FASTA, the pioneer of fast sequence alignment. FASTA, which stands for Fast All, uses a brilliant heuristic strategy. Instead of trying to align the entire sequence right away, it breaks your query sequence into small, overlapping chunks of a fixed length, which we call k-tuples. It is like looking for a unique two-word phrase in a book's index rather than reading the whole book. FASTA scans the database for exact matches of these short k-tuples, creating a map of hotspots. By connecting these hotspots, it performs detailed alignments only in promising regions, drastically reducing the search space. Now that we know FASTA looks for hotspots, how does it mathematically connect them? It uses a simple but powerful metric called the offset. The formula is C equals I minus J. Here, I represents the index position of the k-tuple in our query sequence, and J represents the index position of the matching k-tuple in the database sequence. The variable C is the offset. Physically, this offset represents a specific diagonal line on a comparison grid. If multiple matches share the exact same offset C, it means they lie on the same diagonal. This indicates a continuous region of similarity without any gaps. As I and J increase together, C remains constant, signaling a strong alignment. Before we move on, let us see this offset formula in action with a concrete example. We are given a short query sequence, ACTGAC, and a database sequence, TGACGT. We want to find hotspots using a k-tuple size of 2. First, we break both sequences into overlapping two-letter chunks. We find our first exact match for the chunk TG, It is at position 3 in the query and position 1 in the database. Subtracting these gives an offset of 2. Next, we match the chunk GA at query position 4 and database position 2. This also gives an offset of 2. Because both matches share the exact same offset, FASTA recognizes this as a continuous matching region, serving as a strong seed for further alignment. So we have seen how FASTA uses exact matches. This brings us to BLAST, the most widely used bioinformatics program in the world. If FASTA is like looking up an exact phrase in an index, BLAST is like using a modern internet search engine. When you misspell a word slightly online, the search engine still knows what you mean. BLAST does something very similar. In nature, biological sequences often undergo mutations, where one amino acid is swapped for a similar one. Because BLAST does not demand perfectly identical short chunks right from the start, it can find distant biological relatives much faster and more accurately than older methods. Now that we understand why BLAST allows for variations, let us look at how it actually works. BLAST improves upon the speed and sensitivity of FASTA by using a neighborhood word approach. Instead of just breaking the query into exact words, BLAST generates an expanded list of all possible words that would score above a certain threshold when aligned with your query. It then scans the database for exact matches to this expanded list. When it finds a match, which we call a seed, BLAST attempts to extend the alignment outward in both directions. This extension creates what is known as a high-scoring segment pair, or HSP. Let us visualize this process with a diagram. At the top, you see the original query sequence, broken down into short words. Notice how arrows point down to a central box labeled neighborhood words. This represents BLAST generating that expanded list of similar, high-scoring variations. From there, arrows point down into the large cylinder representing the massive sequence database. The points where these neighborhood words hit the database are our exact matches, or seeds. Finally, look at the horizontal arrows extending outward from those seeds. This shows the algorithm extending the match in both directions to build a high-scoring segment pair. This visual perfectly captures how BLAST filters massive databases down to meaningful alignments. This brings us to the most important mathematical concept in BLAST, the expectation value, or e-value. Once BLAST finds an alignment, we need to know if it is a real biological relationship or just a random fluke. The formula is E equals K times M times N times e to the power of negative lambda s. Here, E is the number of alignments we expect to find purely by random chance. The variable M is your query length, and N is the database length. K and lambda are statistical constants. Finally, S is the raw alignment score. Physically, as your score S increases, the exponential term shrinks, driving the e-value towards 0, meaning the match is highly significant. Conversely, a larger database N increases the chance of random matches. Now that we have the formula, let us calculate an e-value together. We are given a query protein of length 100 and a database of 10 million amino acids. Our raw score, S, is 50, with constants K as 0.1 and lambda as 0.3. First, we substitute all these values into our equation. Multiplying the constants and lengths gives us a massive search space factor of 100 million. Next, we multiply lambda and the score to get an exponent of negative 15. The term e to the negative 15 represents the extremely small probability of getting this score by pure chance. Multiplying this tiny probability by our massive search space gives a final e-value of approximately 0.03. Because this is much less than 1, it represents a true biological relationship. Following our calculation, let us dive deeper into what this e-value actually means for a biologist. An e-value of 0.03 literally means that in a database of this massive size, we would expect to see a match this good by random chance only 0.03 times. That is roughly a 3% chance of a fluke. Because this value is much less than 1, researchers can confidently conclude that the alignment is statistically significant. This rigorous statistical foundation is the true power of BLAST. It tells scientists exactly how much they can trust their results, perfectly balancing incredible speed with the ability to filter out the noise of massive databases. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that FASTA pioneered the field of fast sequence alignment by using exact K-tuple matches to quickly identify promising hotspots. Then, we saw how BLAST took this a step further by introducing neighborhood words, allowing it to find distant evolutionary relatives despite genetic mutations. Remember that both are heuristic algorithms. They trade mathematically perfect, exhaustive searching for the immense speed required in modern biology. Finally, we explored the e-value, which provides the rigorous statistical proof needed to confirm that an alignment is a real biological relationship and not just a random coincidence. You now understand the fundamental engines driving computational biology.